So what am I going to speak about? So I'm going to take you through some of the demographic trends. I'm hoping there's some geriatricians in the audience who will be familiar with these. And I'm also going to talk about variability and malleability of aging. So like Robert, I also hit 60, in fact, last month, so I'm getting more and more interested in this process. I'm going to focus then a little bit on features of immune aging, so what happens to your immune system as we get older. But then I'm going to really move into how much of what we think of the aging process, focusing on immune aging, but I'm happy to speak about other aspects, that's really a result of our modern lifestyles. And then moving on to how we can do something about this, what we know about immune aging and the mechanisms involved that have led us to improving immunity in old age. Then I'm going to finish with a little bit of MedSci life data on myself. So this is why I've become interested in aging and why we've got an Institute of Inflammation and Aging at Birmingham. So these are UK statistics, but it's pretty well similar wherever you go in the world. So if you look at the under 16s, the birth rates are falling, but it's the over 65s that are increasing in their number because we're living longer. So in that 25-year period from 1984 to 2009, we acquired another 1.7 million over 65-year-olds. But the group that are in fact increasing at the fastest rate are these groupings in the dark blue. These are the over 85s, the oldest old. And in that 25-year period, their numbers doubled to 1.4 million. And the predictions are that by 2034, almost 1 in 4 of the population will be over 65 and 5% will be over 85. But there's a lot of variability. So this is data for Birmingham. So longevity is also highly variable. So this is our cross-city line, which today isn't functioning because Storm Doris has thrown trees all over the line. But just 8 stops on this line from Snow Hill out to Four Oaks here. There's a 9-year difference in life expectancy. So here we are in the 21st century, and we've still got big differences in life expectancy. And you'll find this in most cities in the UK. But our experience of old age is also highly variable, and I'm going to introduce you to 3 European ladies, all actresses, and in one picture they're 21, and in the other picture they're 78. And we begin with the Italian representative, Sophia Loren. So here she is, 21 and 78. And she's now actually 81. She's still acting. She was in a film directed by her son just last year. So I think a great example of aging successfully and healthily. Now we move to the French representative. This is Brigitte Bardot, 21 on the right and 78 on the left. And Brigitte is now also 81. She was born just a couple of weeks after Sophia. And she might have got a few more wrinkles, but actually she's still very active. She's politically active. And so I think another great example of what old age can be like. Now we move on to the British representative. This is Elizabeth Taylor, 21 on the left and 78 on the right. And Liz never made it to her 79th birthday. She passed away just a few months after this photo was taken. And she spent the last years of her life wheelchair bound, really not a successful old age. So longevity is variable and your old age is variable. And here's some of the data on life expectancy. So female life expectancy increasing, and it's increasing at 2 years per decade. Put another way, a child born today has a life expectancy of 5 hours more than one born yesterday. It's very dramatic. But this is why we've really started to pay attention and why the government has paid attention. Around 25 years ago in most countries in the developed world, healthy life expectancy started steadying off. So health span is not keeping pace with lifespan. And on average, a female is unwell for the 19 years of her life at the end of life, and a male it's 16 years, with, of course, significant impact on our NHS, and I don't have to tell you about this. It's all over the news continuously. So we need a definition of ageing. Ageing is not just a number. It's not just adding another candle to your birthday cake each year. And this is the one that I use. So it's the increasing frailty of an organism with time that reduces your ability to deal with stress, resulting in increased chance of disease and death. And I'd ask you to bear that in mind as we go through the talk. So, how are we going to move forward? If you think of the current paradigm in disease, all of these diseases here are age-related, so they increase with age. And we currently try and tackle them one by one. So if we're trying to cure cancer, we do cancer research. cardiovascular disease, we do heart disease research. But if you think if all of these are actually caused by the aging process, they're age-related, why not just tackle this process? And I hope I can persuade you this is possible. So, here's one example, caloric restriction. In this technique, what you do is you take any sort of species, and these here are all responsive to caloric restriction, and you cut their calorie intake by 25 to 30%. What happens when you do that is that the animals live longer. So here's the ad libitum fed C. elegans. There's the calorically restricted. And they live on average about 25% longer. So reducing your calorie intake will get you to live longer. You're probably thinking, yes, but that's lower species. What about higher species, Professor Lord? What happens there? Well, it turns out it looks as though it's true there too. These are studies in the US, and there are 2 of them ongoing in macaque monkeys. And at the moment, these macaques are 33 years old, so they're equivalent of about a 75-year-old human. So here's the ad libitum fed macaque there on the left, and you can't see from where you are, but he's got cataracts, he's lost most of his teeth, the poor guy's lost most of his hair, curvature of the spine here. This is an aged monkey. Look at the guy on the right. This is the calorically restricted monkey. No cataracts, he's got most of his teeth. This guy will give you a really good bite if you get anywhere near him. Lovely straight spine there, kept all of his beautiful ginger fur. This is a really spunky monkey. This guy's up for everything. But we don't know yet whether they're going to live longer. They probably are. But look, this is what's the really interesting thing. When you look at the instance of all of the age-related diseases, so cancer, cardiovascular disease, diabetes, neurodegeneration, in the ad libitum group, you've got only about 20% that are disease-free. In the calorically restricted group, you've got 70% are disease-free. So it looks like they're going to live longer, and they're certainly going to live healthier. But you're probably thinking, but those are macaques, Professor Lord. This can't possibly be true in humans. Of course, the answer is we don't know yet. The longest caloric restriction study in humans is only 8 years old. But if we look in the world of epidemiology, we get some clues that it may be true for humans too. So these are data from Okinawa, an island off the southern tip of Japan. And Okinawa has the highest fraction of centenarians in the world. They have 18.5 per 100,000. The nearest to them is mainland Japan, that has 4.5 per 100,000. So they really have a huge number of centenarians. But observations in the Okinawans have shown that part of their social behaviour is they say that you should leave the table slightly hungry. So they naturally calorically restrict. And when you look at the intake of children, they're actually only taking in 60% of the recommended calories that we state here in the UK. And adults, it's about 80%. So they're living longer, but do they live healthier? Well, yes, they do. If you compare Okinawans with Americans, they're 75% more likely to retain cognitive ability. They have 80% fewer breast and prostate cancers, fewer ovarian and colon cancers, fewer hip fractures if they fall, and 80% fewer heart attacks. So there's a hint there the caloric restriction will probably work in humans too. Okay, so hopefully I've persuaded you that longevity and aging itself are both malleable. What about the immune system? This is what I'm going to lead into now. This is what I spend most of my time working on. So if you think about the role of the immune system, it has to detect and kill pathogens. It should be able to remove damaged, senescent or transformed cells. It should react better on second exposures. That's the basis of immune memory and vaccinations. And it should do all of this without damaging itself. So what's the evidence that your immune system ages? Well, if we look at bacterial infections, so these are just data for C. difficile and MRSA, and I looked at the data from 2007 to 2010, and you can see, particularly for C. difficile, really age-related increase in cases of C. difficile. Not quite so marked for MRSA, but still about a doubling of your chance of getting one of these hospital-acquired infections the older you are. What about vaccination? These are some nice data from Beatrix Grubik-Lobenstein in Austria, but it's true. Here is tetanus, but it's pretty well true of all vaccinations. So above this red line, you've made enough tetanus antibody to protect you should you be exposed to tetanus, and below it, you're less likely to be protected. And you can see immediately after the vaccination, if you're 70 and younger, then the chances are you'll make a protective response. 70 and older, chances are you won't. But look here, 6 to 10 years later, now it declines to 50 and younger. You've still got enough antibody to be protected, but 50 and older, then you're less likely to be protected. And in Austria, they now recommend that tetanus should be boosted every 5 years and not every 10. Other evidence of immune memory declining, of course, is shingles. So you're exposed to varicella zoster when you're young, as to have your chicken pox, you develop immunity to it, but it's not lifelong. The older you get, you lose your immunity and it re-emerges as shingles. So your immune memory also fades with age. Again, I won't have to tell the audience that cancer, if you think this clearance, this immune surveillance role, also compromised by age. So most cancers are profoundly age-related. This is acute myeloid leukemia with strong age relationship, but it's true of most cancers. So that immune surveillance role of the immune system also declines. What about this idea that you shouldn't harm yourself? Well, this is rheumatoid arthritis, and again, incidence increases with age, and several autoimmune diseases increase with age. So that aspect of the immune system also compromised. Lastly, is the aged immune system frail? So is it just susceptibility to infection, or is it more than that? So hopefully these data will persuade you that it is frail. So the vertical bars here are flu epidemics. The green line is patients going to their GPs with a respiratory infection. The blue line is hospitalization, and the yellow line is death. So if you're 45 to 64 years old, the incidence is around 900 per 100,000 in the peaks of the flu epidemic. But in this age group, you're highly unlikely to end up hospitalized, and you're certainly not going to die in that age group if you get the flu. 65 to 74 years old, now we've got an incidence of 1,100 per 100,000, so increased susceptibility as you get older. Now you might end up in hospital, about 10 to 15% chance you'll end up in hospital, but still a fairly low chance of you passing away from infection. Over 75 years old, now we see a big difference. The incidence is now up at 1,400 per 100,000 in the peaks of the infectious periods, and there's now is about a 30% chance you'll end up in hospital or passing away. So you're frailer, your immune system is frailer, you're more susceptible to infections, but as a result of the infection, you've got an increased chance of dying. So why is this? What are some of the mechanisms involved? And I'll take you briefly through these. So we look at immune memory, what's going on there? So to remind you, the naive T cell is produced in the thymus. When it meets its antigen, proliferates like crazy, then controls the infection either through cytolytic T cells killing the virus-infected cell or the production of antibody through B cell help. The majority of the cells then die by apoptosis, but you retain some memory T cells so that you've got then immune memory. What happens with aging is that your thymus atrophies. So this happens in humans from puberty and happens at about 3% per year. So by the time you get to 70 years old, you've only got about between 1% and 5% of the thymic output that you had as a 20-year-old. So you've got less naive T cells produced. But your immune system keeps its lymphocyte numbers level, so you undergo homeostatic proliferation. So you increase the number of your memory T cells. You also increase the naive T cells that are in the circulation as well. But overall, you get a change in your naive to memory ratio. So you've got more memory than naive T cells. And if you think of this in evolutionary terms, this makes perfect sense, because what you've done is you've created a cassette, a memory cassette, to the pathogens that you will meet on a regular basis. And as long as you live long enough to pass your genes on, hey, who cares what happens after that? So it makes a lot of sense. And you think, well, this is great then. You know, what's the problem? But, of course, I probably don't have to tell you that your lymphocytes can't proliferate forever, like most cells in the body. They have telomeres. And every time your cell replicates, you lose 50 bases of your telomere, and eventually the telomere is so short that you get then cell senescence. Lymphocytes are unusual. They've actually got telomerase, so they can extend telomeres, but this doesn't last forever. And this is shown here in data from Arnie Akbar at UCL. So he looked at CD8 telomere length in young and old healthy subjects. And you can see in the older patients, subjects, they had much shorter telomeres. And when he looked at lymphoproliferative syndrome in younger patients, they also had shorter telomeres. So the more your T cells proliferate, the shorter your telomeres are. And this then affects the ability of the T cells to proliferate when it's exposed to the antigen that it was previously adapted to. So to summarize that, you've got less new T cells being generated. You're acquiring senescent T cells, which don't proliferate well at all. And you're, through homeostatic proliferation, increasing your naive and memory pool. But it gets even worse than that. So you've got your memory T cells. They're not great at proliferating, but these highly differentiated cells also lose a co-stimulatory molecule, CD28. And you may remember that what normally happens in your immune system, that you have to have two signals. So you have peptide presented on MHC, and then you need this co-stimulatory signal. And the antigen-presenting cell is primed by a danger signal by exposure to a tissue injury marker or a pathogen. And normally, if that's missing, then you get anergy. What happens in these highly differentiated T cells is that they begin to express natural killer cell receptors. So things like KLRG1, NKG2D. This allows them then to get the second signal from self cells, from tissue damage markers. So this means that you're more prone to autoimmune disease. And we showed this in a study a few years ago. So if you look and compare young and elderly healthy subjects and just look for rheumatoid factor, so this is your anti-IgG antibodies, then these are increased even in the healthy elderly. And you find a whole range of autoantibodies present in healthy elders. And one of the reasons involved, we looked at these regulatory cells, the B regulatory cells, and we find that their induction is much reduced with age. And when you stimulate them either directly through ligating CD40 with CD40 ligand, then this is affected with age. And when we looked into the reason behind it, we find the defect there is largely there in the B cell itself. So the signaling through this molecule STAT3 is profoundly reduced in the older person's T cells. So your neutrophils, so you're more prone to bacterial infections, your immune memory fades, and you're more prone to autoimmunity as we get older. But what about this aspect? What about the frailty? So I've explained why you might be more susceptible to the infections, but why is the older patient more frail when they do get an infection? So for this, we really began to think about the innate immune system. And we've focused a lot of our work on the neutrophils, but I can talk about monocytes as well. So to remind you, these are the most abundant white cell. To carry out their function, they have to leave the circulation, chemotax toward the site of infection. There they fight phagocytose, the target, and then they have a range of bacterial killing mechanisms. They can generate reactive oxygen species intracellularly and kill intracellularly. And we now know that they can also kill extracellularly. So they can release their nuclear DNA extracellularly. This is then peppered with proteases and histones that help to immobilize the bacteria and may even in some cases kill the bacteria. So what happens with age? So this is my earliest foray into the world of immune aging. So we showed early on that in response to E. coli, even in healthy elders, that the ability of the phagocyte to uptake bacteria is reduced by about 50%. So all of the neutrophils of the older subject can uptake E. coli, they just eat about 50% less, which is not great if you've got a bacteria reproducing every 20 minutes. When we look at killing mechanisms, so this is net generation. So on the right here, you can see this is staining for DNA. So the bright rings here, these are the neutrophils that haven't released their nets, and the more diffuse staining is where they've released their nets. And whether you stimulate with an artificial stimulus like PMA or CXCL8, the elderly donor neutrophils release less nets, so their extracellular killing mechanism is also reduced. Why is this? Well, this net generation requires reactive oxygen species to stimulate, to trigger the whole process. And so reactive oxygen species, this intracellular mechanism, is also reduced in the older adult. So that helps to explain why perhaps the adult is more susceptible to infections as you get older. But again, what about this frailty aspect? So to remind you, when your leukocytes move towards the site of infection, what they do is they actually release proteases into the matrix, and literally I think of them as burrowing through the tissue. So we decided to analyse chemotaxis. To use this, we use a thing called an insel chamber, so you can place the neutrophils in one side of the chamber and the chemotractant in the other, and then using time-lapse video microscopy, look at their migratory tracks. And you can look at the overall speed, chemokinesis, the directional speed, velocity, persistence, so how often they change direction, and from that give an overall chemotactic index. And this is what it looks like for a young, healthy subject. So it starts here and ends here. Beautiful straight line. The CXCL8 is at the top in your viewing here. This is an older subject. I don't think you have to be an expert immunologist to see that there's a problem here. The target's at the top. Look, this one's going backwards, sideways, they're changing. They've lost their sat-navs. So this is a healthy elderly person, so this is problematic. And it's not just a chemokines. When we expose the neutrophils to sputum from a patient infected with pneumonia, we see this same age-related defect. So when you've got quite a complex chemotractant mixture, the neutrophils still can't migrate really well from the older adults. When does it start? Well, I'm sorry, Robert, it kicks in quite early. So it begins actually in midlife, but it's statistically significant by the time you get to your 60s and 70s. So a true age-related effect. But as I said, why we think this is important is because of this. This is the vascular endothelial cell with the neutrophil there, and they create this area of damage as they migrate through tissue through this release of neutrophil elastase. And so we worked with some mathematicians to model what might happen, and these models predicted that the rather rambling movements of the older person's neutrophils would cause about 40% more damage as they move through the tissues. This would then be 40% more inflammation. So we had to test this out. So we worked with respiratory physiologists to figure this one out, and they developed this assay for a peptide that's released when neutrophil elastase breaks down fibrinogen in the matrix. And this peptide accumulates in serum, so you can measure it quite easily. And when we looked at our healthy old versus healthy young subjects, it wasn't 40% higher, but it's actually double. So these neutrophils are causing the damage the whole time. And this correlated very nicely with higher levels of systemic inflammation. And this is well recorded in old age. Even in healthy elders, they have a higher level of systemic inflammation. It's termed inflammaging. But you're probably thinking, but when they've got pneumonia, the neutrophil will be primed and they'll behave wonderfully. No, it actually gets even worse. So my colleague Liz Sapey, this is the comparison of the healthy young and the healthy old, the chemotaxis. But when you look at lower respiratory tract infections, community-acquired pneumonia, and sepsis with pneumonia, actually it gets even worse in the older subjects. And when you get an extreme situation like sepsis, even the younger person's neutrophils don't cope very well. So at times of infections, the neutrophils have even lower chemotaxis. And when you look at this is data. So now this is the data for that little peptide that's released in a young patient with pneumonia versus an elderly patient. It's now 5 times higher with much higher inflammation. So we think this is the reason behind some of this frailty with old age. Okay, so that's a little bit of what happens to immunity in old age. What about this idea of how much of it might be related to modern lifestyles? So this is just to remind you that this increased longevity is a relatively recent phenomenon. So until around 150 years ago, average life expectancy, unless you were royalty, was 30 to 40 years old. The increase for royalty actually began around 1750. But it was around 1850 that the rest of us started to live longer. So relatively recent. So if you think about this again in evolutionary terms, have we really had chance to adapt to changes in lifestyle? So how much of what we think of as aging is actually lifestyle? Food is much more readily available than when we were hunter-gatherers, and we're a largely sedentary society. So if you look here, some of the data for the UK, these are data for the males on the left and females on the right. So these are the guys that are pretty sedentary. These are the guys doing moderate activity, and the dark blue are those meeting the chief medical officer's recommendations for physical activity, which is 150 minutes of aerobic exercise a week. So in males, it's around 5% of the over 65s are meeting it, so very low. And ladies were even worse, it's 1 to 2%. So we get more inactive as we get older. So how much of this might be driving some of the aging phenomenon? Well, again, a lot of epidemiological data suggests this might be the case. So this is some beautiful data from Katie Corr's analysis of the EPIC cohorts. So you can have a little tot up on your hands in the audience. So you get 1 point if you're a non-smoker. Another point if you have moderate alcohol intake, so 1 to 14 units a week. Another point if you meet the physical activity guidelines, 150 exercise per week. And another point if you have 5 portions of fruit and veg a day. If you score all 4, you've got 14 years longer life expectancy. So if you only score 2, try to get 3. If you've only got 3, try to get 4. Okay, so we wanted to test this out a little bit further. So we want to address this idea of how much aging might be lifestyle related. We decided to look in people who'd kept to what we would consider to be a good lifestyle, highly active adults, and we picked cyclists. We picked cyclists because we reasoned that it's a good aerobic exercise. They would also have good balance, so otherwise they'd be falling off the bike. So you can't have runners like myself because we might have rubbish joints by the time we get to 50 years old. So we thought they were a pretty good group to work with. And like most people who are keen on their exercise, they're actually quite nerdy. So they keep records of what they do. So we could actually verify that these guys had been as active as they claimed to be, because they all had cycling logs. And so we had 125 cyclists, 84 males and 41 females, and we picked them at 55 to 79 years old, so that the ladies had gone through the menopause, so we were going to look at aging. And to be in the study, they had to have been able to cycle 100 kilometres in 6.5 hours. For the males, 60 kilometres in 5.5 for the females. They had to have done that twice in the last 3 weeks, so they were maintaining this activity. So these are not Olympians, but they're the guys out in Lycra at the weekend that you'll see around the village lanes, etc. We got them into the lab, so we did this work in collaboration with King's, Steve Harridge at King's. They had a full physiological phenotype, and we took blood and muscle biopsy samples. So this is cross-sectional at the moment, but we're going to do the 5-year sampling next year. What did we find? So if you look, they've got males here and females, and we've split them into 5-year breaks. So if you look at things like body mass, doesn't really change. So we always think that when we get older, we put weight on. That didn't happen in these guys. That won't surprise you. Percentage fat didn't increase. Again, we all often say as you get older, you increase your adiposity. And your fat-free mass, the amount of muscle they had, didn't decline with age. And again, probably won't surprise you, these are active people. But this idea that we lose muscle, sarcopenia, we put fat on, I would argue that's lifestyle related. What about other things? Again, we'll often say that you increase your insulin resistance with age. No, the home IR value didn't change with these people at all, so they maintained good metabolic health. But it doesn't protect you against everything. So VO2 max declined across the groups, FEV1 lung function declined, and maximum heart rate declined. So it can't protect you totally. Some things are intrinsic aging processes. But what about immune aging? So what I've told you so far might not surprise you, but this certainly surprised us. So thymic output, I told you that the thymic shrinks from puberty. So we've got a measure here of thymic output. This PTK7 marker is a marker on recent thymic emigrants. And here's our healthy young. These are some healthy old. These are the older people who have got no comorbidities, but they're largely sedentary. They're not meeting the physical activity guidelines, and they're not involved in regular exercise. And then our master cyclists. It looks like they haven't undergone thymic atrophy. They've maintained their good thymic output. We look another marker, TREC copy. We were trying to figure out why this might happen. We know that in humans, what happens to the thymus is it actually becomes infiltrated with fat. And there certainly was an association with the amount of body fat that the cyclists had, but not an association with the amount of cycling they did. So they might maintain it because they're not getting this fatty infiltration. But we think the much stronger signal is probably coming from cytokines. So some of the cytokines driving thymic atrophy are TNF and a lack of IL-7. So if you look in healthy young and healthy old, we have this decline in IL-7 with age, and we get an increase in TNF, this inflammaging. And what we find in our cyclists is they maintain their interleukin-7 levels, and they don't increase their TNF. So two of the signals that would affect thymic atrophy are not seen in the cyclists. This other one, KGF, is another marker that affects thymic atrophy, but that didn't change. Anything else happening on? So when we looked at regulatory B cells, again, healthy young, sedentary old, and our master cyclists, they also don't seem to have this drop in regulatory B cells. With age, you also see an increase in Th17, pro-inflammatory T cells, and that didn't happen in our cyclists either. So we think they've got probably a reduced propensity to have this inflammatory response and a reduced chance of autoimmune disease as well. But again, it didn't protect against everything. So markers of senescent T cells still increased in our cyclists. So it protects against some things, but not others. Okay, but can we turn the clock back? What can we do about this? How is all this knowledge going to take us forward? And this is where I've done a lot of work with my clinical colleagues. So I'm a basic scientist, I'm not a clinician. And to do a lot of the work that I'm now going to tell you about, I've worked very closely with clinicians. So this is not our data, but to show you that exercise might also be able to be an adjuvant in vaccination. This is some data from the US, from Marion Cahoot's lab. And what they did was they got older adults to go to aerobics classes for 3 months before they had their vaccination response. And you can see that in the ones that did regular exercise, they got a better vaccination response. What about that caloric restriction? It's probably never going to catch on as a lifestyle in any big way. If you think of the current obesity crisis, trying to get people to reduce their calorie intake by 25% is a big ask. But a lot of the drug companies now are trying to make caloric restriction mimetics. We know how caloric restriction works, the pathways that it hits. So here's one of them, rapamycin, that you may recognize as an immune suppressant used a lot in transplantation. And what you do with rapamycin, it's one of these agents that can extend lifespan in several species, including mice. And what Novartis have done, they've done a 6-week trial giving rapamycin for 6 weeks to patients at different doses, and they looked at their vaccine response. And they get anything from a 20 to 30% increase at even low doses of rapamycin. And this works by hitting the mTOR pathway, which is also an aging pathway. Probably, again, not going to catch on. There are lots of side effects, as you can imagine here, but they are now working on rapalogs, so analogues of rapamycin that won't have the side effects. Also think about flagellins. We know from the immune system when you analyze monocytes that one of the toll-like receptors that detects pathogens and danger signals, many of them are affected. They're reduced in either their expression or their signaling, but the one that isn't is toll receptor 5, TLR5, which recognizes flagellin. And so this is a nice study published in Vaccine where they've conjugated flagellin to the HN1 in the influenza vaccine and got a much better response if they include flagellin in the adjuvant. We took a different approach again to this. So again, you're probably very familiar with that circadian biology is one of the sexy topics at the moment. And we know that your body works better at different times of the day, and this includes your immune system. We've done some studies looking at things like thymic output with the time of day and knew that there were more naive T cells in the morning. So we did a very simple study vaccinating people either at the morning GP surgery or the afternoon GP surgery and then looking at the response. And what we found was a very significant difference between the morning and the afternoon vaccination. And what we're now doing, funded by the MRC, is seeing if this also works in Pneumovax. So this is a T-dependent vaccine, but we're also seeing if it works in a T-independent vaccine. And we'll be starting that study in the autumn. Okay, last little bit. One wonky neutrophils. What can we do about those? If they're so important, we've got to get to the basis of this. And again, this is study work I've done with Liz Sapey, who's a respiratory physiologist. We had to understand why they were wonky. So, neutrophil migration is very complex, but one of the signalling pathways that's important for directional control is PI3 kinase. So we wondered if the PI3 kinase signalling was defective in neutrophils from older donors. And so we did the study, and at the top here is the phosphorylation of the P85 subunit of PI3 kinase, which can be a marker of its activation. Control here, and then exposed to different chemokines. And you can see this is within 5 minutes, a very rapid activation of this pathway. But if you see in the old, we see no activation of the pathway. But for the alert people in the audience, what you should spot is the differences that the control, so the unstimulated neutrophils, the PI3 kinase was already phosphorylated. So we wondered, well, if we inhibit this kinase, will we then correct the migration defect? And sure enough, that's what happened. So neutrophils have 4 isoenzymes of PI3 kinase, and if we took either a PI3 kinase delta or gamma inhibitor, we could increase the chemotaxis response. And the great thing is there are now PI3 kinase delta inhibitors in clinical trial. We're not actually following this, we're actually repurposing a drug that hits the same pathway to get the same effect. Okay, so in summary so far, for the science side, I hope I've persuaded you that innate and adaptive immunity decline with age, that lifelong physical activity protects against some of the aspects of aging, but not all of them, and that exercise and pharmacological approaches are being developed to improve immunity in old age and, in fact, general health in old age. So about some of the other stuff, I was asked to talk about some of my career to date, some of the things I thought I might have done differently. So I like this definition of career that you can find, a headlong progression, usually downhill and usually out of control, and that certainly describes the early phases of my career. So I'll be honest, what I loved in the early stages of my career, I just loved being in the lab. I didn't think about much else. So the early stages, I did my PhD at Aston, I then returned to Oxford for my postdoc, and I actually worked in diabetes in the early days. There's no pressure, you just went home at night, you could think about the science, but you were just a data generator, the lab head got the money in for you, you wrote the papers up together. It was a great life. And then reality struck. I was 31, I'd done 2 postdocs, I had a temporary lectureship at Birmingham, I'd been in 3 different departments, 2 different universities, so I'd ping-ponged backwards between Oxford and Birmingham for several years. I got a mortgage. I was on yet another short-term contract. I got a young child and no cunning plan. So what did I do? First thing, it's actually the last thing. I actually got a mentor, or actually say my head of department imposed himself upon me. So it's Ian McLennan, so Robert will know him. And he was super, and he gave me a real reality check. He said, Janet, where's your 5-year plan? Where are you heading? I had to think, did I want to stay in academia? Yes, I did. I pretty well knew that. But what did I have to do to achieve that? I had to make sure I was publishing good papers, and luckily, that wasn't the problem at the time. I was publishing okay. I'd gained some teaching experience through that 1-year lectureship, but I needed to up my national and international visibility, so Ian really said, any opportunity, if you get a chance to review grants, review papers, be on a grants panel, take it. And that was really good advice. So even quite early in my career, I was actually asked by the BBSRC to be on one of their grants panels. He said, you've got to get a personal fellowship, Janet. You've got to show that you can do it by yourself and survive. And again, I was fortunate. I got a Royal Society fellowship that allowed me to set up my lab. And they were great. They really let you get on with things, didn't pester you too much, and it was a great experience. Working abroad is the one thing I didn't do. That's my one regret. And I think about, life balance, I should have done that earlier on because, with the family, it was then much harder. So that's my one regret. And other than little months here and there, I haven't had an extended period abroad. I think getting the right location. So when I returned to Birmingham from Oxford, I'd originally only intended to be there for a year, and that was 30 years ago. And what's kept me there is that my research is really what is now called translational research. And I knew early on that's what I wanted to do. As a basic scientist, I wanted to be doing things that were close as I could get to patients. And this is where I'm located now. I'm in the new Queen Elizabeth Hospital, which sees 1 million patients a year. It's the largest hospital in Europe. And we're actually integrated into the hospital. We've got basic labs here. We've got our own clinical research facility so that we can literally translate from bench to bedside very quickly. And the institute is about half clinicians, half basic scientists, so we're interacting constantly. And I think that's an important thing as well. What about the other stuff? What about my MedSci life? Well, hopefully you're all decided you're going to be academic clinicians anyway, but it is a great life. There's lots of stresses there, but some fun things too, so you get to meet some famous people. So the older members of the audience will recognise Sir Cliff Richard, so he came and helped us with a lot of our fundraising, so he's been great. You get to be on the telly. So I was on the telly last year with Angela Rippon in a programme called How to Stay Young, doing a thing called the Sit to Rise test, and our head of college, Dave Adams, routinely comes up to me and just says 10, because that was the maximum score you could get, and he's very pleased that he got the maximum score. But try to keep a balance, so keep your hobbies. So I'm a keen runner. I don't run as much as I used to, so I haven't run a marathon since 2010, but I still try and get out as frequently as I can. I live on the Trent and Mersey Canal, so I try and get out on my boat as often as I can as well. Keep other hobbies as well. So this is scouting. So until 2 years ago, I was Arcaler and had been an Arcaler for almost 40 years. So I'd done this since I was doing my GCSEs. So I think it's good to keep that balance. The only thing that stopped me was needing to get to the scout hut every Wednesday at 8:30. So I could no longer do that. But I still go out and help on the camps and things like that. So try and keep a balance. Keep the fun in your life. Don't make it all work. I think that is important. And you can get involved in political campaigns even if they don't succeed. So this was our effort to try to show that science really is something that flourishes when we work together and we don't have national and international boundaries imposed upon us. So hopefully we'll try and keep that going. And I think that might be it. This is my wonderful team at John Hazeldene's wedding. It really is teamwork, and I think you'll all probably develop and be members of teams, and I've enjoyed very much being involved in teams that are a real mixture of basic scientists and clinicians. So Liz Sapie, I couldn't have done any of my neutrophil work without her. Mark Fosteris, who is a trauma surgeon. He's actually a military trauma surgeon because Birmingham's also the Royal Centre for Defence Medicine, so I do my trauma work with him. Chris is a burns surgeon. Daisy's a trainee at the moment. She's doing a PhD with us. Thomas Jackson is a geriatrician. And the 3 people in yellow are the basic scientists who did the work. So thank you for your attention.